So this is sort of where we left off. And this is a tree that shows you this part of Opisthocons within Unicons, right, that has these choanoflagellates that people have, for a very long time, suggested were closely related to animals. Then we have sponges, which you'll notice they're showing them here, as sponges is not a real thing. It's a paraphyletic group at the base of animals. And then you have all of the other animals. You'll see the name we're going to use for all of the other animals is Eumetazoa. Eu meaning true. They're the true animals, the animals that have true tissues. Sponges, I think you could actually make an argument that sponges maybe aren't really true animals. That brings up an interesting question as to where you decide what is an animal. Assuming this is the correct phylogeny, notice that you could decide, and most biologists probably would say, you know, this is where animals start. So sponges are animals, all the eumetazoans are clearly animals, but choanoflagellates are not real animals. The reason Whittaker pulled choanoflagellates out is because they're unicellular. And he said, you know, well, things like paramecia and amoeba and all these things that are unicellular, if you're not multicellular, you can't be a real animal. That's what he said. Obviously, though, things like paramecia and amoeba are not even related at all, right? You all know that paramecia are what?
Alveolata.
They're alveolates, right? And amoebas are amoebozoans and stuff. They're clearly not even related to animals, right? But choanoflagellates are very closely related to them. So you know what? If you just ease that rule a little bit and say, you know, animals don't have to be multicellular. They could be unicellular. Then you could decide to draw the line out here and say everything here is an animal. And then choanoflagellates are just unicellular animals. Likewise, I think you could say, hey, you know what? If you don't have true tissues, we're not going to consider you a real animal. And so we're going to say that animals start here, and that this group is animals, and these are just closely related groups, right? But they're not true animals. So where exactly you decide to define animals is sort of arbitrary. This is where most biologists would probably put it. So you know, on an exam question one time, I asked my students, much to my dismay, I said, you know, some biologists might argue that sponges are not true animals. If they're not true animals, what are sponges? And do you know what like half my class put? Well, if they're not animals, I guess they must be plants. What? Why would they write that? This is towards the end of the course. What are plants? Plants are archaeplastids, right? They're related to green algae and red algae. They have nothing to do with this at all. What I was hoping they would say is, well, you know, maybe if they're not really animals, maybe they're sort of like colonies of individual cells. You'll see that there's no true tissues here. There's no muscles. There's no nerves. This is basically like a highly organized set of little individual cells that just all kind of live together. But you'll see there's no gut. There's no central vascular cavity that they all share anything. Instead, each little cell is sort of feeding on its own. So that was what I was hoping they'd say, is that it's an elaborate colony of choanoflagellate cells or something like that, not a plant. Right? Yeah. Maybe. You all would put plant. This is the picture out of Brooker. When you looked at, do you remember in the lab you were supposed to take those prepared Grantia slides, those sponge slides, and you were supposed to look for the individual sponge cells, the choanocytes, right? Choano means collar. You'll notice that these cells have a little collar and a little flagellum. The flagellum beats and draws food particles into the cell. If you look at a choanoflagellate cell, it looks a lot like this, right? I mean, it's not a mistake that biologists for a very long time, before molecular phylogenies or anything, thought that choanoflagellates were closely related to animals because they look a lot like one-celled sponges in a way. If you actually looked for those cells and tried to draw them, you probably realize they were real small and really hard to see. Notice the textbooks checking out, they show you a cartoon because it's very hard to see these things. They're extremely small. Do you see them here? This picture was taken at the University of Chicago. We went out on a field trip and collected some pond water and stuff. This is a single cell from a green alga. Here's the cell wall between two adjacent plant cells. These are individual choanoflagellates. There's their little flagellum. Do you see it? That's at like 1,000x. Look at how tiny those are. And you don't see any beautiful collar or anything. You can barely see that there's a flagellum there or anything. These are very small. But if you looked at those sponge cells, they're really tiny, just like this, right? And basically, this is what a sponge is. If you took a whole bunch of those cells, put a little bit of an extracellular matrix, add in some spicules so it has some skeletal elements, there you go. That's a sponge. It's basically a whole bunch of these cells working together in a little colony. But it's not really a tissue in the sense that every cell is feeding on its own. Every cell is living its own little life, right? It's really a whole bunch of cells living together. It's not a whole bunch of cells that are working together sort of in sort of a concerted way that we think of like as a tissue.
Does the function of the flagella still for locomotion?
No, because they're sessile, so they sit in one spot, right? Like those little guys don't get up and swim around or anything. They sit there and they beat the flagellum basically and draw food particles into the collar, and that's how they feed. So if you had Campbell's 5th edition, you're all working with either Campbell's 8th edition. We didn't adopt the 9th edition, we switched to Brooker. So you either have Campbell's 8th edition probably, is that right, for those of you that have Campbell, or you have Brooker's whatever edition that we're on. So this would have been the phylogenetic tree for animals, the animal kingdom, in Brooker's 5th edition, right? And there are probably about 30 or 40 animal phyla. You realize we're not going to cover 30 or 40, not because they're not all super cool, but because this class is just sort of a survey class, so we're going to hit kind of the big 8 or 9. So things like the peripheral, we're going to treat sponges as though it was a phyla, maybe not. Cnidarians, ctenophores, platyhelminths. We're going to kind of skip things like rotifers, nematodes, nemertians. This week's lab focuses heavily on these guys, mollusks and annelids, right? This is a huge, 2 huge major phyla that we're going to focus on this week in lab. Arthropods, another just massive phyla. We're going to focus on that next week. We're going to kind of skip over bryozoans and phoranids and brachiopods. And then the week after that, we'll have this really big lab on echinoderms and chordates, and then we go to MOCA and kind of look at all of them. You'll notice in this tree from Campbell's 5th edition that they sort of have these things that have only a single opening to the gastrovascular cavity, that have a blind gut, things like cnidarians and platyhelminths. Those are sort of towards the base of the tree. You'll also notice that there's a bunch of emphasis on this idea as to whether or not you have a coelom. Y'all remember what that is, right?
Cavity.
It's a secondary body cavity that houses internal organs. So if you're acoelomate, you don't have a coelom at all, that's sort of a basal ancestral condition maybe. And then the animals like nematodes and rotifers that have pseudocoeloms, right, that develop from the blastocele. What's the difference between a pseudocoelom and a true coelom? A pseudocoelom is not completely lined with mesoderm, right? It touches endoderm in certain places. So that's sort of a little bit better, but not really as good as a true coelom, right? If you have a true coelom, that's by far the best. Ends up this is completely wrong. This is the way it would have been presented there. Lots of emphasis on coelom type, which ends up being a highly convergent trait in animals, so you can't use it to classify them this way. A little bit of attention to this protostome versus deuterostome. And you'll notice here an interesting relationship, this relationship between annelids and arthropods, which for the longest time were thought to be close relatives. And the reason is because they both have segmented bodies, and that segmentation was thought to be something that indicated close relatedness. I'm going to show you a picture from this invertebrate zoology book, that one that I like so much from the 80s, that shows a polychaete worm, like the one we have in lab tonight, an onychophorid velvet worm, and a millipede. And you'll see they just look exactly alike. These perfect little segments, little appendages coming off each segment, cute little head, little antennae, cute little eyes. I mean, they look exactly like one another. You could easily imagine how a nereid worm would have somehow become a millipede. They look exactly the same. It ends up that the nereid worm that looks so much like a millipede is actually much more closely related to snails and clams. And there's an awful lot of evidence to support that now. But for almost 200 years, this idea that arthropods and annelids were closely related to one another persists. It's wrong. There's a lot of evidence now to support that that's really not right. So this is Brooker showing you the old version. And notice again, there's a fixation on coelom and pseudocele, and segmentation is a character that unites annelids and arthropods. This ends up being not good characters. And about 10 years ago, maybe 15 now, the first molecular phylogenies for animals came out. And they were radically different than this. And the zoologists that looked at it were like, wow, this can't be right. There's just no way. Because you'll see in a second, arthropods are actually most closely related to things like nematodes. That can't possibly be right. This is like telling you that a penguin's more closely related to a zebra than it is to some other bird, right? I mean, it sounds like complete ludicrousy, right? It sounds completely impossible to imagine. Biologists, zoologists, when they first saw the tree that I'm going to show you in a second, they were just like, that can't possibly be right. It's got to be wrong. There's got to be something wrong with the way that they're doing the analysis or something. But over time, they've come back and they sort of re-examined what they thought they knew about the biology. It ends up that the determinate cleavage pattern that you see in things like arthropods is actually probably the same determinate cleavage pattern that you see in things like nematodes, and different from the one that you see in annelids and mollusks. It ends up that there's a larval stage that's shared by mollusks and annelids that you never find in arthropods. It ends up that arthropods and nematodes shed their outer covering. Annelids and mollusks don't do that. And it also ends up that, for example, the pseudocele that you see in a nematode, maybe the schizocele. If you go back and look at Campbell's 5th edition, they're going to talk about how arthropods have a schizocele. Notice it doesn't say that in Campbell anymore. Now it says annelids and mollusks. It doesn't include arthropods in there. That's because a lot of biologists have gone back and re-examined whether or not the hemocele in an arthropod really is a coelom that's homologous to the type that you have in annelids and mollusks. This is where molecular phylogenies are at their best, is when they force biologists to go back and rethink and try to reinterpret what they thought they knew. Remember I said we don't know things in science? All the stuff they knew was leading them to a tree that really was not right. And now, over about the last 10, 15 years, they've amassed a tremendous amount of evidence that this is really not on the right track at all, and that something like this is actually closer. Campbell's 6th edition has a whole chapter that does nothing but talk about why the tree in the 5th edition is probably wrong, and this one is probably a much closer guess. You'll notice there's nothing on here about coelom type at all, right? And that's because you have things that are acoelomate, pseudocoelomate, true coelom, pseudocele. It's a mess. It's a highly plastic, highly convergent character. And it was throwing them off when they were trying to base relationships on an understanding of coelom type. Coelom type is a bad, bad character to use. Instead, they look at sort of this, sponge is always towards the base. Again, probably not monophyletic the way they're shown here. Probably the radiata or the radially symmetrical cnidarians and ctenophores are not monophyletic. You might remember that ctenophores actually have bilaterally symmetrical elements to their body, so they're sometimes called biradially symmetrical. Ctenophores also have a mouth and something that kind of at least partially qualifies as an anus. So that whole idea that you only have one opening to the gastrovascular cavity, that's not really kind of right here either. You'll see that we talk about these animals as being diploblastic, which means what? That they have two endoderm and ectoderm, two germ layers, but that they don't have mesodermal tissue. Well, what makes mesoderm mesoderm? There is something that's between the endoderm and the ectoderm. For example, a jellyfish is called mesoglia. So if there's something in the middle, why is that not mesoderm and they're triploblastic? It's because a tissue has to be made up of cells. You can't have just stuff, right? It has to be cellular in order to be considered a tissue. Well, some of the jellyfish, guess what? They actually have some cells in their mesoglia. So some people start to interpret that as being mesodermal tissue. Can you see how nothing's ever as easy as you want it to be? All these rules that I keep giving you, like, you know, all animals have an animal life history. I still think that's right, but it could be wrong, right? All animals are heterotrophic. Somebody showed me that there's an animal that you could clearly argue is autotrophic. So you have to be careful, right? Anytime you think you really understand something, something else will come along and convince you actually don't. But notice we have sort of this major division between the radiata and then what we would consider the bilaterians. Bilateria appears to maybe be a real group. This probably not. And this gets divided into these two major groups, protostomes and deuterostomes. So protostomes, remember, the blastopore becomes the mouth, and deuterostomes, the blastopore becomes the anus. And protostomes, these two newly recognized groups of lophotrochozoans, which I'm going to tell you what this refers to in a second, and ecdysozoans. And notice within the lophotrochozoa are things like platyhelminths. So all of a sudden the platyhelminths that have only one opening in their gastrovascular cavity, don't have a coelom, are up in this highly derived group with things like mollusks that have true coeloms, true schizocele, and annelids that have a true schizocele, and clearly all these guys have an open gut. All of them, I think, except for the platyhelminths. In the case of the platyhelminths, like tapeworms, they don't have a gut at all, right? They've lost their digestive tract completely, but why is that? These guys have highly specialized life habits, especially flukes and tapeworms are parasites, right? Why would you need a gut if you live in a place where you're floating and digesting food all the time? You just absorb it through your skin. There's no reason to ingest it and then try to digest it further or something. You just absorb it through your skin. They've lost their gut. These guys probably lost their anus. They're nested up inside of a group that all have a complete gut. That's probably the ancestral condition here, is to have a complete gut. They've lost their anus. They may have lost a coelom. Who knows, right? But all these characters that we're looking at these guys and interpreting as being ancestral are probably derived, right? Just because something's simpler, you realize like snakes don't have legs, not because they're representing some kind of an ancestor. They're descended from animals that had 4 legs. They lost their legs. Yeast are not unicellular because they're ancestrally unicellular. That's a derived condition for yeast. They're descended from fungi that are filamentous, right? And they've reverted back to unicellularity, right? So you've got to be very careful when you're looking at a simple body form and interpreting it as being the ancestral condition. It might not be. It might be a derived condition, and that's part of what really sent people on the wrong path with this for a long time. You'll notice here, though, annelids are lophotrochozoans, and arthropods are these ecdysozoans. You'll notice that they're split apart. This was one of the most troubling things for most zoologists when they looked at this, because everybody knows that annelids and arthropods are closely related because they both have segmented bodies. And like I said in that book, they even lay it out. Annelid, Nereus worm, polychaete worm, onychophora, and millipede. They actually show it like it's a transitional form or something. Like there's concrete evidence that basically these guys gave rise to these guys or something like that. And that's really not right at all. So we have these sort of two newly recognized groups within protostomes, and then we have the deuterostomes, which have sort of long been recognized as a group. They've kind of been recorded.
So is animals a monophyletic group?
And animals is a monophyletic group. Yeah, animals is a monophyletic group. Either all sponges plus all eumetazoa, or all eumetazoa by themselves, or even if you included all the quinoflagellates, you could make animals monophyletic. Yeah, animals is monophyletic. The problem is groupings inside of animals, our understanding of relationship. Most of the phylum, you'll see in a second that platyhelminths, you have to pull one group out in order to make it monophyletic. So it's polyphyletic the way it's historically defined. But most of the animal phyla, like mollusks, the way that mollusks were historically recognized, gastropods, bivalves, and cephalopods, that's monophyletic. Annelids, polychaetes, oligochaetes, and hirudinans, that's monophyletic. So most of the animal phyla, most of the animal classes as historically defined. Notice I've slipped back into Linnaean ranks now. Did you notice up till now I wasn't saying phylum, class, order, and now all of a sudden I am, and you're like, wow, this guy's crazy, why is he doing that? It's because in animals it works. So all of a sudden I make believe that we're talking about the animal kingdom, and I'm going to talk to you about the phylum Mollusca, the phylum Annelida, the phylum Cnidaria. The reason is because most of those phyla within animals are, for the most part, monophyletic. They mostly work. What we were very confused on is what's the relationship between the phyla. That's what I'm showing you in these trees, right? All those names that are up here, like this, Porifera, Cnidaria, Platyhelminthes, Rotifera, these are all phylum names. So what we were really wrong with is how the different phyla within animals were related to each other. Yeah? But the monophyly of animals is still okay.
But you said that sponges weren't monophyletic, and you included them in the group.
You have to include all of them. Sponges by themselves are not monophyletic. So like, for example, what this is showing sponges as one thing. Really, there's like one sponge here, one sponge here, one sponge here. If you include all the sponges, right, and go back to this ancestor here, all the sponges plus all the eumetazoa would be monophyletic. Yeah.
Okay.
What's not monophyletic is sponges by themselves. Even though they're showing it this way in this tree, but that's probably not right. You're going to learn it that way, but that's probably not right.
When you say pull the group out, what about that group? I mean, is it
I'll show you exactly where it goes. There's actually a little group that's potentially the sister group to the Bilaterians. It's not a lophotrochozoan. It's not nested up inside of the protostome animals. I'll show it to you in the next tree. This is Brooker's version of the same thing. Notice here they recognize this group of lophotrochozoans by either a lophophore, which I'll show you what that is in a second, or a trochophore like the larva stage, and then you have the arthropods and nematodes held together by this character of ecdysis, which I'll tell you what that is in just a second. So here's protostomes, deuterostomes, these are the bilaterians, right? You have true tissues here, right? So sponges don't have true tissues. And then this idea of radial symmetry here. Again, they're holding Cnidarians and ctenophores together, but the most current tree in Campbell, at least, doesn't really do that. So what is a lophophore and what's a trochophore larva? What are the characters that define a lophotrochozoan? This shows you a bryozoan or an ectoproct, right? With a lophophore. This is the lophophore. It's a little crown of tentacles that it uses for feeding. Most of the members of the Lophotrochozoa have either a lophophore or they go through a larval stage called a trochophore larva. If you look at this, it's very distinctive with this little apical tuft of cilia, and then it has this little midrib of cilia. Very distinctive. If you were to look at like a little marine snail or a marine worm, when they go through their larval stages, they make things that look like this. It's a very distinctive type of larval form. There's no arthropod would ever make a larva like that. You're going to look at lots of arthropod larvae, none of them look anything like that. So had they looked at the larval stages of some of the annelids and some of the mollusks, somebody might have gone, wow, you know, they do look a lot like. It's kind of like with Saprolegnia and Laminaria, you know, kelp. If you look at the spores, 2 laterally attached flagella, one smooth, one tripartite tensile hair, you're like, gee, they really do look a lot alike, right? It's all a matter of what exactly you look at. But if you look at just the fuzzy stuff on the dead fish and you look at the kelp, you're like, wow, those don't look anything alike at all. This one's phalloid and it's photosynthetic. This one's filamentous and it's doing absorptive nutrition. It's heterotrophic. Those don't look anything alike at all. But you look at the spores, they're almost indistinguishable. And they're very, very distinctive because you don't see other things that have that type of flagellar arrangement outside of the straminopods. Same thing here, right? If you look at this particular trait of a trochophore larva, you find it within members of this particular group of animals. You don't find it outside of this group. Yeah? This shows, these are the pictures in Brooker. So here it shows some little Lophophoriid that has this little Lophophore, and then shows a picture of what a trochophore larva looks like. So what's this? This is a cicada, I think, and he is molting. That's what ecdysis means. Ecdysis is sort of a fancy word for molting. So these are the ecdysozoa, the molting animals. If you're not familiar with the word ecdysis, you know, if you say that somebody's an ecdysial artist, you know what that means to do? Like, oh, you know that? She's an ecdysial artist on the weekend. What does that mean? It's a stripper.
Oh, nice.
It's a fancy way to say stripper, right? So these are the stripper animals, because they all have some kind of a coating that they shed. In the case of arthropods, it's an exoskeleton. When you look at the ascaris, the nematodes next week, the cross-section, you'll see that they have this thick cuticle, and sure enough, they molt that cuticle. So do onychophorans, so do tardigrades, all the guys that are in this group. Nobody would have ever thought to put them together, and yet sure enough, when the molecular phylogenies pop them together, somebody sits there and goes, oh my God, these guys all have some kind of a thick covering on the outside that they molt periodically. Annelids don't do this, mollusks don't do this. So this group is called the ecdysozoa, this group that unites these things that nobody would have ever thought to put together, like nematodes and arthropods. So here we go, Lophotrochozoa and ecdysozoa. So you actually have a character to put for each one of those, kind of like we had for albulated and straminopod. There's a clear little morphological trait that appears to be maybe a synapomorphy that would actually define the group. Okay, this is the cover of that invertebrate zoology book I like so much. This is from the 80s, you realize, so there's some issues with the classification, but the biology in here is great. Somebody told me they found a copy of this in the public library. She said, don't tell anybody because she doesn't want you to go get it. But there's supposed to be a couple copies in our library. Did anybody go check it out?
They have some on the Venice campus.
And they have one in Venice, at least one, maybe a couple. I love this book, and so I asked them to get it because this is the one that I used like 100 years ago when I took invertebrate zoology. So I wanted to make sure that you kind of knew what it represented. So what do you think this is right here, like these guys? Anybody know what this is?
Bacteria?
They're supposed to be bacteria. Anybody know specifically what this is that has these little chains of cells and then there's like one cell that looks different? Like maybe a Anabaena, yeah. Looks like a heterocyte with the little photosynthetic cells. And this is obviously supposed to be what? So this would have been Kingdom Monera. This would be what? Protista, right? See the little groove here with the transverse flagellum and a trailing flagellum? This is a what? Dinoflagellate. This is a Euglena. This is probably a radiolarian. This is a amoeba. This is a paramecium. This is obviously supposed to be the kingdom what? Fungi. This is a what? Who's that? You know that one? Probably not Aspergillus, but the other one with the conidium? Penicillium. And this is who? Rhizopus. Here's a mushroom. What's this? Yeast. Any idea what this is? I think that's a plasmodial slime mold. It's like Phacerum, right? These used to be considered fungi, right? So 30 years ago, these would have been put in with fungi. How about this? This is obviously supposed to be who? Plants. Who's this? It's a moth, right? There's a little sporophyte with his foot stuck in his mother. See how much stuff you know that you didn't know a few weeks ago? Who's this? Here, with the little umbrellas. It's a little archegonial head, right? With the little sporophytes hanging under there. Who's this?
Liverwort.
It's a liverwort. That's Marcantia. Here's a fern, right? And then some flowers and stuff. Who's this supposed to be? Fish, amphibians, reptiles, birds, and mammals. Who would that be? Vertebrate animals, right? And then all of these guys are invertebrate animals. So now, and you know a few of them, like who's that? Dugacia, right? Here's Lumbricus that you're going to see tonight. Here's a little clam like Donax that you're going to see tonight, right? Here's a roundworm. So now the question is, why do you think it's drawn that way? Why do you think that such a big part of this is invertebrates and just this little part over here is everybody else? Any idea? What does the pie represent, the slices in the pie? Proportions of species described. So if there's like maybe, how many? 1.5, probably more, 1.6 million species described. Almost 75% of them, about 3/4, are invertebrate animals. By far the most of any. I mean, if you look at these other groups, all the fungi in the whole world described, species described, just a tiny fraction compared to these, right? All the plants, all the vertebrates. You probably think, wow, it's lots of vertebrates. You take all the fish, all the amphibians, all the reptiles and birds and mammals. There's only about 4,000 mammal species. It's close to 1 million invertebrate animal names that are described, right? Where do you think most of those are? They're not like flatworms.
Insects.
They're insects. And specifically, what group of insects?
Beetles.
Beetles, right? Coleoperans. That is like by far the most species. If you want to like have work forever, go describe new beetle species. You'll never end. You'll just be describing new species constantly. Somebody asked Haldane, y'all remember Haldane? Who's that? Remember Oprah and Haldane had the idea about abiogenesis and how early Earth-reducing atmosphere might have led to the abiogenesis of organic compounds from inorganic material, right? Somebody walked up to Haldane at a party and asked him, what can you say about God? You know what he said? An inordinate fondness for beetles. God must love beetles because there's more beetles than there are anything else, like 1 billion beetles to 500,000 of everything else. So, yeah, tons and tons of invertebrates. Hopefully you realize that invertebrates is not a real thing. What do I mean when I say it's not a real thing?
Not monophyletic.
Not monophyletic. The word invertebrate doesn't really mean anything in a biological sense. Obviously it means something. It means it's an animal that doesn't have a backbone, right? That's what an invertebrate is. But that's not a real group. Vertebrates, on the other hand, is a real group, right? Vertebrates is a monophyletic group within chordates that we're going to define. It's kind of like non-seed plant, seed plant, right? You're basically grouping all these guys together based on what they don't have, and what kind of a trait is that probably for animals? It's probably a... Go ahead, say it. Somebody start to say it. It's the ancestral condition. It's probably a shared ancestral trait. It's a simplesiomorphy. You can't define a group based on that. It's not going to give you a real group, right? Way back in January when I taught you those terms, you didn't really think I'd still be using them, so I knew them. But see how it keeps coming back. Okay, so these, like I said, there's probably about 30, 40 phyla. We're going to learn the big 8 or 9. So sponges, which we're going to all treat as peripheral, although notice some books are already starting to divide them into like 2 different groups of Calcarea and Selacia, which are individuals that have calcium carbonate spicules versus silica spicules. Apparently that's a division within sponges. Cnidarians and platyhelminths, which you've already all looked at in lab. So my goal is to definitely get through at least the first couple of those. Although I'm moving faster with you guys, might get further. We're going to kind of skip over things like this, like rotifers and lophophoriates. Not that they're not really cool, and ectoprocs and brachiopods, but there's just not enough time to cover them all. You all going to someday take an upper division in vertebrate zoology class, right? You realize I had a student, she left, she went up to USF, she was taking like a 3,000 level in vertebrate zoology class, and she sent me an email and she said, when I was taking your class, I thought you were completely crazy, but every single thing that you went through, that's exactly what we're doing here. Because you will do this if you take an invertebrate zoology class. It'll basically be something like this. These are the guys we're looking at in lab tonight. These guys are cool. Some of my favorite animals that we're going to look at in lab are actually in lab tonight. So those guys are great. These are the animals we're doing next week. Huge lab. This is just a massive group. I just kind of convinced you 2 out of every 3 described species on Earth is in this lab. So obviously we're going to have a challenge getting through that. And then echinoderms and chordates. And chordates, of course, is a big, important group because that's where all the vertebrate animals are. And then we go to moa, right? So where you kind of get to see all of them. Okay, so this is the tree. Which one of you, Joel or somebody just said, well, what happens when you pull it out, right? Here's part of what happens when you pull it out. This is probably a slightly better tree. I think this is out of animal. Notice here, if we say that these are going to be all animals, right? Sponges are not shown as monophyletic. Ctenophores and cnidarians are not shown as monophyletic together. And then we have this group here, which would be the bilaterians, the bilaterally symmetrical animals. So we have the deuterostomes here, like echinoderms and chordates, and then we have the protostomes here, these new groups we've recognized of lophotrochozoans and ecdysozoans. Now remember platyhelminths, flatworms? Most of them go here. All the ones you looked at in lab. So y'all remember the classes, right? Platyhelminthes, Turbellaria, Trematota, Cestoda, right? When I say those names, is there a picture in your head? Like when I say Turbellaria, are y'all picturing that cute little Dugassia that you were looking at? When I say Cestoda, you're all picturing a tapeworm. You're going to somehow attach a picture to these names, right? Most of them, most of the flatworms go in here, right? But there's this one weird little group that were used to be considered platyhelminths, right? That are called aceals. Very, very simple little flatworms. It ends up that they actually appear not to be related to the other flatworms, but instead, they appear to be potentially the sister group to all of the bilaterally symmetrical animals, all of the other bilaterally symmetrical animals. That makes these guys rock stars. Nobody would have even cared about them. I never would have even mentioned them if we were doing this a few years ago. But the most current molecular phylogenies show that these might occupy the sister position to all of the other bilaterally symmetrical animals. Why is that so important? This is the holy grail, you realize, in phylogenetics, is to find the sister group to arguably one of the most important groups of organisms on Earth. You're talking about every single animal, basically, except for jellyfish and sponges, right? Why would it be so cool to know who the sister group to that is? Because the sister group gives you the best idea as to what this might have been like. It gives you the best insight because it branches off. It's occupying a branch that's most close, presumably, to the last common ancestor that they all shared. So if you ask me, what did the last common ancestor shared by all bilaterally symmetrical animals, what did it look like? What was it like? What was its biology like? My best guess would be that it was something like this, based on this tree. There's another one that's not shown here. There's a weird little animal called a placozoan, which is about 1,000 cells with almost no differentiation, a little ciliated underside, and it just kind of moves along. People thought that it was a really highly derived, weird cnidarian. It now appears to occupy the sister position to all of the Eumetazoa. So it would be tucked right in here. I'm going to show you a picture of it in just a second. If you have Campbell, it treats these really well. I'm sure Brooker mentions it too, although I have to look back at Brooker again. But I'm sure it mentions placozoans and aceals, right? Placozoans is potentially the sister to all the Eumetazoa, and aceals as potentially the sister group to all of the bilaterians. Yeah? This is sort of a summary tree. Start putting this together in your brain. Remember how before you'd go, what do I need to know? And I'd show you that huge eukaryotic tree that would have excavates and alveolates and stromatopiles and rhizarians and archaeplastids and uniconts, and you somehow kind of got that. Did you all get that, kind of? This is where you need to start putting this. This is how you need to start grouping animals. So you need to be able to take these phyla and somehow put them into this general framework, because this is sort of the phylogeny that we're going to work off of. So this is a sponge. This is a, well, it's bigger than a hydra. It's a sea anemone. What phylum is this? Cnidaria. What class is this? Does this sound remotely familiar to you? Yes, because you already did this in lab. What class is this? These are the flower animals. How do you say flower? Anthozoa, right? That's where sea anemones and corals go. This is a jellyfish. What phylum is this? Obviously it's the same phylum as that, right? Cnidaria. How about, what class is it? Scythozoa, right? The one that I don't have pictured right here would be Hydrozoa. That would include Hydra and Obelia. Physalia. Who's Physalia? You realize it's like taking German or something, right? Is there a picture in your head when I say Physalia? There's no picture. That's the problem. It's a Portuguese man of war. You all remember the Portuguese man of war? He was there in the class. It was there in the class. How about what's this? I showed you a little picture of these guys zooming around. We're going to have live ones at Mode, I hope. There was a little dead one in the lab. These are comb jellies, right, or ctenophores. They're in a different phylum from Cnidarian. These do not have staining cells, right? And remember, these actually have that anal pore. I don't know whether it's really an anus, but they have an anal pore, so they kind of have a one-way digestive tract. They're biradially symmetrical. They have these 8 ciliated comb rows. No nematocysts here, right? Presumably diploblastic, but no nematocysts here. And remember, they have like a cytipid larval stage, right? What's the larval stage for Cnidarians? You know, I'm going to do it in a second. Call it planula larva, right? A lot of them go through a polyp stage, which is part of the larval development. Any guess as to what this is? That's a placozoan. That's the thing I just told you is a superstar, because it's potentially the sister taxon to all eumetazoans, to all the true animals. If you ask me what the last common ancestor to all true animals look like, not sponges, but everybody else, I'd guess this. Might be wrong, but based on what we know right now about animal phylogeny, this would be a reasonably good guess. About 1,000 little cells, ciliated underside, just slides along. Nobody had any idea what this was. Single species in this lineage. Never would have talked about it before, and all of a sudden now it's like this super important thing because it might potentially be the sister group to all the eumetazoans. And guess what these are? Aseals. So that's what you were asking me, Joel. What happens when you pull them out? When you pull them out, they become superstars because now all of a sudden they're occupying the sister position to all the bilaterally symmetrical animals. That's where they went. So they're aseals. They're in a phylum on their own, really, if you want to think of it that way. These guys are touted as having idiosyncratic determinate cleavage. I wouldn't call it that. It's got to be some other type of determinate cleavage other than the type that you see in the ecdysozoa, which we're going to call them determinate, but that's the way they can be. So these actually have a determinate type of cleavage. These have radial cleavage. But the ctenophores, I should have pointed out, have some kind of a determinate cleavage pattern too. Determinate cleavage evolved multiple times. These are the guys we're covering in. So those were the guys we covered in the lab last week, as much as we're going to. These are the guys we're covering this week. This is a rotifer. We're not going to cover rotifers. So you only have to know them in this picture. That's it. This is a bryozoan. What does bryozoan mean? What does bryo mean? Like bryophytes, who were those? Mosses. It means moss animal, right? Ectoproct is another name for this group of animals. Guess what? We're not covering those either. How about this? Don't say clam. Those have absolutely nothing to do with clams. Clams are mollusks. They go down here. These are brachiopods, lamp shells, they're sometimes called. They have nothing to do with clams at all, right? We're not covering them either. We're not covering any of these guys. There are 3 animal phyla that will go in here. We're not covering these either. These are ribbon worms. Hopefully you realize the word worm doesn't mean anything biologically. Everything we call a worm has nothing to do with anything else we call a worm. These are Nemerdians. Nemerdia would be the phylum name for these. These are the ones I really want you to know within the Lophotropozoa. These guys are all Lophotropozoa, they're all protostomes, but these are the ones I want you to know. This is a, what do you think that is? What phylum is it? Flatworm. We already covered this. These are platyhelminths, right? Yeah. What class would those be in? They're in the same class with Dugesia. You all know who Dugesia is, right? That little planarian that you looked at in the lab with the 2 little eyes that look like they're crossed on his little head. You were trying to feed him egg yolk, remember him? This is loosely related to him, except this is a marine version. This is not a freshwater version. Notice it gets bigger, and notice they tend to be very colorful. These are the ones I'm going to show you penis fencing in the movie. So these are platyhelminths. These are annelids, or segmented worms. Kind of looks like a millipede, doesn't it? I have these guys live up in the lab for you. They're the coolest things imaginable. This is Nereus. This is a little Christmas tree worm. That's actually the little tentacle part of it. The actual worm is down below, right? So some of these make little tubes, and they actually live in a little tube with their little head with tentacles sticking out one end. Earthworms go in this group. Leeches go in this group. I have all these live in person for you upstairs. You're all planning to go play with a leech, right? Lots of segmented worms in this group. And then mollusks, which are going to be gastropods, things like snails and slugs. Bivalves, what do you think those are? Clams, bivalve, 2 shells is what their phylum name means. Clams, oyster, scallop, mussels, right? And cephalopods, which are going to be octopus, nautilus, cuttlefish, squid, which you're going to dissect tonight. This is lab this week, yeah? And notice, really, these guys should have gone into the lab this week. The reason I put them in last week's lab is just because this week's lab's got a lot of stuff, and last week's lab didn't have any stuff. Next week's lab are the other group of protostomes. So that was one side of the protostome group. This is the other side, the Ecdysozoa. Who are these? The guys that mold, the stripper animals, right? So this is a nematode or a roundworm is the common name. Remember, these have a pseudocoel. These are two phyla that we're not going to really cover. These are,
Lice.
huh?
Lice.
No, lice are actually insects. They're arthropods. These are, the common name for these are water bears. They're tardigrades, right? Very tiny little guys. Velvet worms, these are onychophorans. And then arthropods, which is this huge group that includes everything from arachnids, like horseshoe crabs. You realize horseshoe crabs aren't crabs, they ruin your day. I'm sure you're getting used to this by now when I go, raspberries are berries, strawberries are berries, prairie dogs aren't dogs, right? Pineapples aren't apples, right? Horseshoe crabs aren't crabs, sorry, they're not. Horseshoe crabs, and I'm going to convince you that's very easy to show you. Once we start counting appendages on arthropods, they have a body plan that's very much like that of a spider or a scorpion, ticks, mice, horseshoe crabs, kind of all go into this group that we're going to call the chelicerates when we get there. Crustaceans, which unfortunately is not monophyletic unless you include insects, so things like crabs and crayfish and lobster, right? All of the insects, which is just a huge, massive group. And then the ones that aren't really shown here would be things like millipedes and centipedes, obviously, are in there too, right? Myriapods.
Going back to the horseshoe crab, you were saying that's an insect?
No, I'm saying insects are crustaceans. Unless you include insects within crustaceans, crustaceans as historically defined is not monophyletic. So all the things that we would have thought of as crustaceans, even though you'll see in your book, it's actually going to treat, it's going to have these four subphyla. It's going to say chelicerates, and it's going to say myriapods, and it's going to say crustaceans, and it's going to say insects. But insects is actually inside of crustaceans. Crustaceans is not monophyletic without putting it inside. Horseshoe crabs, though, you'll see, so whereas crabs, like true crabs, are in the old group Crustacea, and they're in this group now that we're going to call Pancrustacea, in a class Malacoster, I think we're Decapoda. I mean, a crab is a crab. These guys are not crabs. The horseshoe crabs are not crabs at all. They're actually, they have a cephalothorax and abdomen. They don't have all these different segments that you have in a true crab, and they're not going to have all these different appendages. They just have two preoral pairs, chelicerae, and then they have, second pair is not really highly modified to pedipalps, and they have 4 pairs of walking legs. You'll see that when you do a crab, crabs have 2 pairs of antennae, then they have a mouth, and they have mandibles, 2 pairs of maxilla, 3 pairs of maxillipeds, 5 pairs of walking legs, and then these swimmerettes and uropods, tells and everything. It's just a totally different body plan in a true crab than what you would see in a horseshoe crab. And you're going to do this because you're going to flip over a spider, a scorpion, and a horseshoe crab, and you're going to compare them, and you'll see the body plan is very, very similar. When you realize what I just did where I named all the appendages there, you realize you're all going to be able to do that, right? Because that's on the next lab practical. I wouldn't know that if I didn't teach in this course. I studied mushrooms, right? So you're all going to be able to do that. But you'll see, it's very easy to distinguish them. And of course, phylogenetically, horseshoe crabs are not related to insects. And then the deuterostomes. So this is the last lab before we go to moat. And this includes some groups like hemichordates. This is an acorn worm. Can you see how there's worms all over the place? Basically, you're on a skinny or a worm. These aren't related to one another in any way. Echinoderms, which are going to be things like starfish. You're going to dissect a starfish, sea urchin, brittle stars, sea cucumbers. You'll realize that's an animal, right? So if you go to like a Chinese restaurant, you order sea cucumber, that's not a cucumber. You're eating an animal related to a starfish, basically. And then this huge important phylum called Chordata, which includes a few invertebrate animals, like this little tunicate here, but it also includes all of the vertebrates. So all of the things that we call fish, which you'll find out that the word fish doesn't mean anything either. Amphibians, reptiles, that's what you were all going to call this, right? You all think of penguins as reptiles now, and mammals, right? So a very big group that we're going to go very quickly through, yeah? Okay, so you all ready? So what I'm going to do now is start plowing through the file. Porifera, Cnidaria, Platyhelminthes, Annelida, Mollusca, Nematoda, Arthropoda, Chynodermata, and Chordata. And all the detail for each one. And then we take the test, and we're almost done.